Hey everyone, welcome back to the AI Research Roundup. I'm Alex. It's May 5th, 2025. Today, we've got this ICLR conference paper, Proteina, published just over 3 months ago. This paper presents a really large-scale flow-based model. That's a way to generate data for creating brand new protein structures, de novo design. It tackles scale and control issues using a big transformer and new conditioning techniques. Okay, so this first figure really visualizes the core idea. You see how it starts with a cloud of points, basically random coordinates, like Gaussian noise, and uses this flow-matching technique to gradually transform them into a realistic protein backbone structure. The big transformer model learns this flow, and notice that optional input? That's the hierarchical fold class label for conditioning, giving that fine-grained control over the final structure. Building on that, this next figure shows some really impressive results. Actual long protein samples generated by Proteina. We're seeing structures from 300 all the way up to 800 residues long. Quite large. And notice how some are guided to have specific features, like more beta sheets, showing that conditioning we talked about really works. It highlights the model's power in handling scale and control, a key advance here. Okay, so this figure dives into the data behind Proteina. Part A really shows the scale. Look at the D21M dataset, over 20 million structures. That's enormous compared to previous ones like PDB. Then, Part B visualizes those hierarchical fold class labels used for conditioning, C, A, and T levels. This massive, richly labeled dataset is absolutely key for training such a large model and enabling that precise control over generated structures. Alright, now this figure really unpacks the engine driving Proteina, the transformer architecture itself. It shows how the model takes in the sequence information, those conditioning labels we just discussed, and also pairwise features between residues. You see the main processing stack there in Part D, using adaptive multi-head attention. This whole setup is specifically designed to handle that massive scale and effectively integrate the conditioning for precise control over the generated protein structures. So this figure provides some concrete examples of that conditional generation in action. Using those hierarchical CAT fold class labels we discussed, the model can generate specific structures like Tim barrels, Rossmann folds, or even target mainly beta sheet content. Crucially, these generated samples are designable and actually match the requested fold class. It really demonstrates the precise, fine-grained control Proteina offers over the protein structures it creates, which is a major step forward. Okay, moving from those specific examples, this table gives us the hard numbers, comparing Proteina's unconditional generation against other methods. Look at the metrics: designability, diversity, novelty, and those new ones like FPSD and fold score. You can see Proteina variance, particularly M_21M, trained on that huge dataset, achieving really high designability, like 99%. This really backs up the claims about its state-of-the-art performance, especially showing the benefit of scaling up the training data. Following up on those overall performance numbers, this figure specifically zooms in on how well Proteina handles generating long protein backbones. You see two plots here: designability and diversity versus the number of residues. Notice how Proteina, the blue line, maintains really high designability and good diversity, even as the length increases up to 800 residues. Well, most other methods really struggle and drop off much sooner. This clearly shows Proteina's strength in tackling larger, more complex structures. So, wrapping things up, we've seen how Proteina really pushes the boundaries, especially in generating those very long, complex protein structures where others falter. And that fine-grained control using fold labels, well, that's a huge step too. It truly establishes a scalable, controllable foundation model for protein design. Okay, that's all for this AI Research Roundup. Thanks for tuning in. I'm Alex.